Good afternoon, everyone. My pleasure to welcome you to a very special lecture today. This is the annual Margaret Pittman Lecture, in which we invite a distinguished woman scientist to come and address the NIH community and recognize this by the name that's attached to this lecture, Margaret Pittman. She was NIH's first female lab chief. Margaret Pittman made significant contributions to microbiology and to vaccine development, particularly in the areas of pertussis and tetanus, during a long career in the National Institute of Allergy and Infectious Diseases, from which she retired in 1971. In fact, when Margaret Pittman began working at NIH, it wasn't here in Bethesda. It was down there in Washington, and she moved with it back many decades ago, but is recognized as having really been a pioneer both in science and particularly in the place of women in science. We at NIH feel this is a really important issue and have a whole committee that looks at these issues regularly, and we're very happy to be able to highlight particularly today the Margaret Pittman Lecture by bringing in a spectacular speaker for you to listen to, Dr. Paula Hammond. Dr. Hammond got her undergraduate degree in chemical engineering at MIT. There'll be an MIT theme here. She did then get a master's degree in chemical engineering at Georgia Tech, but then returned to get her PhD at MIT and then joined the faculty, where she has been continuously since then, beginning as assistant professor and now as the David Koch Professor of Engineering and chair of the department at MIT. And that's the Department of Chemical Engineering. Her research, which you're going to get to hear a lot about, is groundbreaking in terms of its potential for medicine as well as a variety of other applications, because it is in this general area of nanotechnology, and particularly the self-assembly of polymeric nanomaterials, with a major emphasis on the use of electrostatics and other complementary interactions to generate functional materials, with lots of applications in medicine that I think we will all be pretty excited to hear about. Dr. Hammond, in addition to her academic achievements, is an elected member of the American Academy of Arts and Sciences, is a Ovarian Cancer Innovator Award recipient, has a TED Talk, which if you enjoyed today, you should go and listen to as well, because it's very well done, presenting really quite challenging technology to a general audience and doing a beautiful method of doing so. So delighted that you are all here. Welcome to all of those watching also remotely over the web. And please join me now in welcoming the Pittman lecturer, Dr. Paula Hammond.
Thank you so much. It's a real honor to be able to give this lecture today, especially given the history of the Pittman Lecture. And I've had an incredible time getting to know a number of old friends and meeting a number of new friends and colleagues while I've been here. Now I'm going to talk today, and I think this is my pointer. I'm going to talk today about a layer-by-layer assembly approach, which can be used to generate new methods of delivering drugs. And the nice thing about this approach is that it can be used to deliver drugs on surfaces on almost every length scale, from nanometer length scale up to large macroscopic millimeter and centimeter length scales. This technique is very simple. It involves the alternating adsorption of charged species. Essentially, you can start with a substrate. It can be any substrate, plastic that has been treated so that it has a negative charge, for example, a metal or metal oxide, which has its own native charge. And you can take that substrate and immerse it into a dilute aqueous solution, which contains something that is multiply oppositely charged. Now, when you do that, you're going to get adsorption of that charged polyvalent material onto the surface until ultimately the surface charge gets reversed. And at that point, electrostatic repulsion prevents any further deposition on the substrate. So you have something like a monolayer that is controlled. It's a self-limiting adsorption. And the thickness of that layer is dependent on what you're adsorbing, if it's a polymer chain or a nanoparticle, for example, and the pH and ionic strength of the solution that you're adsorbing from. So you can make these films thicker or thinner based on those kinds of conditions. Once you've absorbed to the point of reversal, you can rinse off anything that's not truly absorbed on the substrate and immerse it into another solution, again, oppositely charged, and that material system will absorb. And we can continue this alternating plus and minus to generate these multi-layered thin films. Now, I show these as green and blue stripes, but in reality, when they're polymeric material systems, they're highly interpenetrated. However, you can get definition within plus or minus 4 layers, which means that if you create a series of layers which have one composition and cap them with a series of layers of another composition, you can create either gradients or systems which have 2 or more delivery systems combined. Now, we can use hydrogen bonding to generate these systems as well. For example, you can combine something that has a polyethylene oxide, which is a common biologically used polymer, with some proteins. So I showed you 2 beakers, but in reality, in our lab, we don't have students who have to move back and forth with their wrists and generate these thin films. Very fortunately, we can use robotics to generate these films very effectively, and we can program those robots to generate any kind of film layer sequence that we want. But even in that process, it can take anywhere from a few minutes to 15 or 20 minutes for each absorption step, which is wonderful for lab, but isn't great for manufacturing. However, this is a very scalable process because you can actually use other methods. And here we're showing one method, which is spray layer by layer, that was an automated form which was generated in our laboratory, in which you can use convection to spray dilute concentrations of these different systems onto a substrate directly, spray the rinse, and then spray the other system. And this is actually a homemade automated spray layer by layer system built by one of my former students who later went on to start a company which made these micro sprayers and then went on to generate large-scale layer by layer systems which can generate 50 feet per minute of huge yards of layer by layer materials. This demonstrates the fact that this is scalable, but spray layer by layer has also allowed us to coat a broad range of substrates, everything from very highly porous substrates to flat ones. Here we can show a cartoon of an electrospun mat, which has micron scale fibers throughout. And if we take a cross section, we can see that we've coated everything along the surfaces of these mats. Here you can see that there's a nanometer scale coating on this fiber, for example. We can change the process conditions to create condensed thin films on these porous substrates as well. And this gives us a huge amount of versatility when we're generating something like a tissue regenerative scaffold. So I'm going to describe 3 stories of how we use this layer by layer approach to generate thin films for a range of different delivery applications. But I'm going to first start with why we're doing this in the first place. The reason that layer by layer is particularly compelling is because we're using water, which means that we can incorporate a range of drugs which are sensitive to solvent and sensitive to heat. Biologic drugs, proteins, antibodies, a range of siRNA, DNA, mRNA, mod RNA, a range of different systems which you normally would not put in solvents and heat up. On top of that, if we think about a more traditional polymer like polylactic glycolic acid as a carrier, this polymer would allow you to put in about 1 to 2 weight% of a material system like a drug. Then the rules of thermodynamics take over and you're going to get phase separation because all polymeric systems can only contain small amounts of another material before you get phase separation. And if you get phase separation, you're going to get a bolus release, everything coming out at once. On top of that, if you put 2, 3, or more drugs into a conventional coating, they're all going to come out at once. On the other hand, with layer by layer, we can select a degradable polyion, like this polycation. This is a family of polybeta amino esters. The main point here is that they have positive charge and an ester group, which allows them to break down, and they're biocompatible. If we use something like this and layer it with a drug molecule that is charged, we can create thin films in which the drug is one of the components of the film. Now we've gone from loadings that are 1 or 2 weight% to loadings that are in the 10, 20, 30% range and can be as high as 50 weight%. This means that we can get a huge amount of the drug into a very small film. And this film is small enough, thin enough that it can coat a range of different substrates and fairly complex geometries. On top of that, we can pair each drug with a different polymer and get a different release rate. Now we're actually tuning the release rates of the drug. If we're able to introduce separations between these drug layers, we can prevent interdiffusion of the drugs during the construction of the film. And with that, we can get not only different release rates for each drug, but a kind of controlled stagger or stage-wise release of each drug. And there are several reasons why this is compelling. One can imagine, for example, the desire to release small molecules from an implant surface, which may address pain or infection, followed by larger macromolecular systems like growth factors, proteins, that can allow or enable the regeneration of tissue. So with that, I'm actually going to describe these three different areas of work. And the first one is about bone regeneration. Here, we're interested in being able to use this technique to essentially introduce growth factors, which normally are introduced in products in high concentrations. One of these is BMP2. BMP2 is osteoinductive. It actually recruits the native stem cells in bone marrow and allows them to differentiate into osteoblasts. What's nice about this protein is that it's potent. It actually has a very high osteoinductive effect. What's been the downside of it is that when it is used in the way that it's typically available, a collagen sponge containing milligrams of BMP2, there's a product, for example, that allows a bolus release of this material, you'll get a huge amount of it released into the bloodstream and only a small amount that resides in the site, at the site of delivery. So in these cases, you end up flooding the bloodstream and the body with BMP2, and you can get ectopic bone growth. You can activate nascent tumorigenic cells and essentially activate cancer. And there are a number of other negative impacts of having large amounts of this molecule. And yet, it's a very promising molecule for treatment. So can we deliver this in a physiologically relevant quantity and over the time periods that are relevant, not in a few minutes or seconds, but over an extended time period? We're also interested in growth factors that can support vascularization. And some of these include VEGF, which is a direct angiogenic factor, and platelet-derived growth factor, PDGF, which is mitogenic, but also supports the generation of the vascular system. So these are proteins that are similar. You notice that they all have an isoelectric point of 8.5. That means that in water at pH 7 or neutral pH, they're positively charged. So we treat them as a positively charged entity. We adsorb them from very dilute solution onto these substrates, and we alternate them with a negatively charged species. The negatively charged species can be polyacrylic acid, which is a very simple, highly charged molecule, or it can be a glycosaminoglycan or a polysaccharide, all of which are nicely negatively charged. And finally, we're going to introduce a positively charged degradable biocompatible polymer. Here we've been using our polybeta amino ester, or PBAE layer. And we call these poly 1, poly 2. We were running out of names, as you can tell, in our lab when we named them. But they give us an idea of how long it takes for the materials to release. In my first example, I'm describing the work of my student Nisarg Shah. In this work, the idea was to see if we could address large bone defects, these critical defects that cannot heal, and replace what is the known therapy, which is to do an autologous bone harvest at a secondary site, usually the hip. This secondary site often is the cause of morbidity, infection, and a range of other problems. On top of that, a number of patients who are suffering from either a large traumatic blast or shock or may be experiencing some form of disease may not be able to donate very much bone. So there's a problem with this. There are also problems with other approaches. We can look at other materials that can sort of cover or essentially protect this area, but then we have an issue with the material interface that is generated as a result of that. So we decided to investigate the release of growth factors over a very small, with very small doses over a very controlled time period from a scaffold that degrades. And to do this, we started with PLGA, which is a known FDA-approved polymer, and we generated porous membranes from them using a very simple processing technique. So these end up feeling like white, soft, floppy films that you can cut out with a cookie cutter, which was done here. And in this case, this was cut to the size of our cranial defect model. We then coated that PLGA, which degrades over a reasonable time frame of a couple of months, with a layer-by-layer film. And here, it's a little dark, but you can see that there's a layer-by-layer film sitting on top of this porous substrate. We can then allow release of the growth factor based on what we've done to incorporate the material. And in this particular example, we built BMP2 containing layers on the bottom and PDGF containing layers on the top. If you remember, PDGF is going to help support vascularization. When we look at the in vitro release of this simple system, we see that there's a slight delay of one over the other. So PDGF comes out over a period of the first week or so, whereas the BMP2 is being released over a much more extended period, which goes out to about 30 to 32 days. Then we looked at what happens when we have single versus dual growth factors, and we also looked at dose. Here you can see micro CT of the bone regeneration process. Here's our untreated control, and you can see the critical defect is, of course, not closing in this racked cranial defect model. However, if you look at the release of 0.2 micrograms of BMP2, you can see that you are getting closure of the critical defect, and it seems to be fairly rapid. With the 2 microgram dose, we've increased the dose by 10, but haven't really changed anything about the nature of the closure of this wound over that time period, which suggests that there is a limit to how much more can be impactful. However, when we look at 0.2 micrograms of BMP2 and 0.2 micrograms of PDGF, with micro-CT, we see perhaps a sign that there's somewhat more rapid healing, but we can't really tell what's going on with the bone until we look at the histology and do mechanical measurements of the native bone and really examine what is being generated here. So here you can see cross-sections from histology of the untreated defect, in which we have this gap that remains, and the margins of the bone. If you just have a PLGA membrane and nothing else, what you get are fibroblasts, which crawl across the membrane and deposit collagen. You end up with this collagenous sort of scab-like material, but not bone. If you look at 0.2 micrograms released from our scaffold, what you can see is that you're going to get bone generated in significant amounts, certainly enough to bridge the entire defect. And this is taken, all of these are taken at 4 weeks. But you also see that the bone is somewhat rough looking. And if we increase the dose by a factor of 10, we simply get a big lump of bone that has very little relationship with the margins. It's essentially a kind of cap of bone that's been formed. However, when we look at the cascade of growth factors, PDGF first, followed by BMP2. If you remember the time scales, that PDGF is released more rapidly and over the first week, and that BMP2 is being released over this period of a month. What we're seeing is as a result is a more vascularized bone that appears to be more like the margins, but we're also seeing more complete remodeling. Rather than seeing a large number of defects and bumpiness at the surface, we see a surface that is very similar to the original cranial surface of the bone. If we examine the bone, look at whether we have trabecular bone or not, we see that we do. We generate very high molecular weight material, high mechanical properties in these materials. And we also are able to, with mechanical measurements, compare them to native bone and see that they match the native bone, unlike what we've generated in the first 2 examples, in which the bone is more brittle and breaks more readily. So we think that what we've done is introduce vascularization earlier in this bone. So this bone is a healthier bone. It's much less likely to become necrotic. But maybe as importantly, we've introduced pathways for a second wave of osteoclasts rather than osteoblasts. And osteoclasts actually remodel bone, and that bone remodeling is what is allowing us to achieve bone which has the mechanical properties that we need. Now, along with looking at the cranial defect, we've also been looking at ways in which we can integrate bone onto implants. And this is actually a growing and incredibly important problem because it turns out that orthopedic implants and their use has increased rapidly. It's in fact because our population has grown older and is healthier, We have more of our population that has a whole joint implant, shoulder, hip, knee. The fact that we're also a more active population adds to the probability of seeing these implants in use. More importantly, there are more revision arthroplasties. That is the replacement of these implants after they wear. Usually they last for about 12 or 15 years. And after that period of time, just the relative amount of loosening that can take place over time can require that the old implant needs to be replaced. We want them to last as long as possible, but we're ultimately going to need to replace this implant over the lifetime of many patients. And what happens in that case is that the infection rate soars from the primary case to this secondary, this revision case, from lower than 1% to, depending on which review article you might read, 10 to 20%. There's a very real increase in infection. So we have a concern about this. On top of that, we would like to generate a very strong bone interface with the native bone and the implant. The better that interface, the less likely that infection is going to return. We are interested in being able to accomplish this using a singular coating because the common treatment right now is a two-step process in which you essentially remove the implant from the patient, you backfill the empty joint area with these antibiotic beads, which release over a period of 6 weeks or more. You brace the patient, you wait, and then you go back for a second surgery. Clearly, this two-surgery extended hospitalization or immobilization period is not only going to introduce more stress on the patient, but also more cost in the system. So in our work, we've been looking at ways in which we could generate an osteoconductive base layer because we're no longer working with a degradable membrane, but with a stiff, rigid material like titanium or PEEK, and we want to make sure that we have an environment on which bone cells can grow and adhere. So in our case, that base layer is going to be composed of a chitosan-wrapped hydroxyapatite nanoparticle that has a net positive charge and will be alternated with polyacrylic acid. We just put down a few layers of this material. We can think of it as a system which doesn't degrade over the lifetime of our release film, but will be absorbed or resorbed by cells over a longer time period. Now we have this base layer that will allow deposition of bone. And on top of that, we're going to place layers of BMP2, which can release over an extended period of time. So this is our early experiment. We haven't introduced the anti-infective yet. We'll get to that. But we first had to ask the question of whether or not we can generate a strong bone material interface. So here we used a rat tibia puncture model, which is demonstrated here. We create a defect in the rat tibia. We take either a plastic or a titanium implant, coated or uncoated, insert it in, and then over time, we are going to observe the bone material interface using several metrics, including histology, mechanical properties, and just general measurement of the concentration of growth factor and other factors in the area. So here you can see the plastic implant that we used, which was PEEK, and PEEK with holes drilled in it. What we did was we coated these two examples, which represent a smooth implant and what we were trying to replicate as a textured implant, was coated with BMP2 that was labeled with a fluorescent protein. So here we can actually see this release over time, and it's a little bit dark here, but you can see that it's releasing over a period of 30 days. If we look at the radiant efficiency over time, we can see that the release is actually quite constant, and it's taking place over this period of a month fairly consistently. We looked at different loadings. 20, 40, and 60 layers are representative of these three different loadings. And we look at the concentration of BMP2 that is accumulated in this area of interest, which is shown here, by excavating the tissue and determining how much BMP2 is there. And what's interesting is the numbers here. You can see over a period of 30 days for the smooth versus the porous or roughened implant, that we can get a more extended or sustained release when we have this texture on the surface or roughening. But what's also interesting is that at any given time, the concentrations don't exceed 200 nanograms per milliliter. And we were talking about the relevance of being able to control the amount of BMP2 released. Here we're getting enough BMP2 in the region to make a difference physiologically without creating enough of a sink of BMP2 to flood the bloodstream with the growth factor. We looked at histology, and I'm only showing you one example, but we have an entire library of histological slices of the tissue. This one is meant to show the interface between a part of the implant, this is the plastic, and the bone. And we can see that the bone is very nicely arranged and oriented around this implant. We actually examined the nature of this bone and found that it was very nicely oriented. It had excellent mechanical properties. On top of that, we did a pull-out test in which we determined the amount of force required to pull the implant out of bone, and we did it at a period of 1 week, 2 weeks, and 4 weeks. Here we can see the untreated control at the bottom. And these implants actually show that there's some bone that is grown near the implant, but it's not integrated into it. And it's very easy to pull these out. But the pull-out forces that we see with these different doses indicated by different numbers show that in the first couple of weeks there's a dose dependence and a very strong increase in the pull-out force. But by the time we reach 4 weeks, everything has leveled off. And we see that even our lowest dose system is able to provide a very highly integrated implant bone material interface. And in fact, the strength of this pull-out force is about 4 times that is generated with polymethyl methacrylate bone cement, which is commonly used. We can also note that when we pull these out, at this point, we see that the fracture takes place in the bone, the native bone, and not at the bone material interface. So now we want to introduce an anti-infective. And the way that we do this is by introducing gentamicin sulfate. Turns out to be another charged drug molecule. And here my student Juha Min came into the picture. She began to first model these systems and their release characteristics and then to build them. Here she's using red to demonstrate the gentamicin sulfate and green to represent the BMP2. And she quickly began to construct material systems in which there was no barrier and you could get some degree of stagger. But she also introduced barrier layers by using naturally occurring lapunite clay. This is just a very thin, disc-like nanoclay. They're used for a number of different applications. In our case, we chose this system because it exhibits good biocompatibility. A number of these laponites are generally regarded as safe, and they're very low cost and biodegradable. They are also potentially bioactive. They can actually enhance cell adhesion at surfaces and interfaces. So what we're going to do is use them as a kind of barrier layer in between the 2 drug layers. The question here is, can we get a stagger that we control? On the left, what you can see is the thickness in the film as Juha built up differing numbers of BMP2 layers. Then she introduced these ultrathin lapunite layers, then gentamicin sulfate, and then in some cases she capped that off with another lapunite layer. The question was whether we could segregate the 2 drugs into compartments. And here you can see with electron microscopy and a method called EDS that these dark regions are the drug-containing layers. These bright regions are the lapunite capping and intermediary layers that separate them. Here we can see red as the silicon-containing layers. These are the clays that are essentially separating our materials. When we look at the release between these different systems, with just the BMP2 and gentamicin sulfate, we get some nice results. We see a rapid release of gentamicin, and we do want enough to eliminate an existing infection, and therefore this bolus release is not bad. And we see that there is a delayed release of BMP2. However, we see that there's a significant amount of BMP2 that's coming out in the first 4 days, where this x-axis is in days, and here we're seeing a blow-up of that release behavior. If we have this barrier layer in between the 2, it turns out that with the BMP2, we actually can decrease this rate of release. We get, in fact, a 14 times longer release period, or half-life. And you can see this in the blow-up a little bit more easily. You've sort of flattened this curve over the first several days while you're getting rid of infection very rapidly with the gentamicin sulfate. And of course, we can tune this even further with the capping layer. Juha was interested in this because she wanted to be able to demonstrate the release of enough gentamicin sulfate in the beginning to eliminate infection, so you have this initial bolus, combined with the ability to maintain this, critical concentration to prevent infection over longer periods of time. So when we then move back to our rat tibia model, now we've generated an infected rat tibia model. So Juha generates a biofilm in this tibia defect cavity. You can imagine this is a rather challenging model, so this was a nice little chunk of her PhD thesis. And she then is able to insert either an uncoated implant, an implant which contains gentamicin sulfate, or one that contains the combination of both. And what we've been observing is that the combination of BMP2 and gentamicin sulfate leads to a much more rapid regeneration of bone around the implant. Furthermore, if we look at the micro-CT, we can see the resorption that occurs in the untreated sample. We can see that with just gentamicin sulfate, bone is enabled. It can begin to regenerate, but it's not quite as complete. And with the combination, we're getting a much more solid slug of regenerated bone present in these systems. And so now our hope is to be able to continue to look at these compartmentalized systems, but to use a much more systematic approach to tuning the relative release of these systems using barrier layers, and also begin to introduce antibiotic combinations or multi-spectrum antibiotics that will give us a broad spectrum of attack, including addressing MRSA. All right, I'm going to move to the second example, which is a soft tissue example. Here, the idea is to try to address wound healing processes that have gone awry. And the idea is that a number of these wound healing processes have overexpression of a specific gene or some sort of genetic dysregulation. Our earliest target was to target chronic wound healing. And the idea was to correct the overexpression of certain genes that are overexpressed in hypoglycemic patients during the wound healing process. So the idea is that we can try and address some of these overexpressed proteins, one of which is MMP9. This is a protease that is a natural part of the remodeling process that all wound healing processes must go through. But MMP9 shows up earlier in the wound healing phase in these diabetic patients. And this has been demonstrated for a number of different studies. So the idea here is to knock down MMP9 for a small period of time and effectively enable the deposition of granulation tissue or collagen, which will then help to lead to wound closure. And I apologize, it looks like there's a little disconnect with my computer and the screen, but what you're seeing here are the same components we talked about before, a degradable polymer, poly2, and here we used chitosan, which is positively charged and not degradable over short time periods, and dextran sulfate, another natural sugar. And the way we composed these films was essentially to build an underlayer of dextran sulfate with this poly2, and then on top of that, layer siRNA and chitosan. And in these studies, we actually used an unmodified version of siRNA, which is interesting because we were definitely meeting the challenge. siRNA degrades very rapidly in the bloodstream, but we're going to put this in a wound cavity where there's a large excess of proteases, cytokines, and other highly aggressive reactive molecules. We directly incorporated siRNA into these films and layered them onto a nylon tegaderm bandage. Here, siRNA is labeled with this fluorescent marker, so you can actually see it in confocal. We're showing that we're coating all of the crevices. We changed the relative number of layers of the underlayer to change the release rate, and the top layers to change the loading. So here we're getting very significant loadings of siRNA in this range of 10 to 20 micrograms per centimeter squared. We then used a diabetic mouse model, and we'll have a control wound in which we don't introduce any treatment. We also have a second control in which we're using a scrambled siRNA, doesn't encode for anything, and we're using our siMMP9 treated system. If you look over time at the gene expression in these animals, at 1 week and at 2 weeks, we were essentially designing these systems to be active over a 14-day period, what we're seeing is a knockdown of MMP9 in the wound of 60% at 1 week and 85% at 2 weeks, which is significant, especially given that these are unmodified siRNA. We also see a corresponding drop in MMP9 activity. If you look at Mason's trichrome staining, we can look at the 7-day and the 14-day treated samples. And here at the top, we have the siRNA treated versus the 7-day untreated. And we also have our 14-day treated and untreated. And what you'll see is there's a bridge of collagen in this stained blue sort of ribbon at the top here that is bridging the wound at 7 days, which we don't see in the untreated samples. And when we get to 14 days, we see that there's a large amount of deposition of collagen present compared to the untreated wound. And here you can see a close-up of that tissue. So there's a stark contrast between the two. And we also see very real contrast in the rate of wound closure in these systems. Here we have a 7-day and 14-day, and the dark bar is our treated sample. And we also see an improvement in the percent epithelial closure in these animals. This is really just the beginning. There is more than one gene involved, of course, in these kinds of wound healing processes. And what we're interested in now is combining targets with siRNA and using growth factors in our combination systems to be able to address this more effectively by replicating the wound healing process. Another area of interest, a real target for us, is trying to address hypertrophic scarring. This is actually something that can greatly influence those who are the victims of third-degree burns, those who have experienced large traumatic blasts. A number of these patients, when they heal with large amounts of hypertrophic or highly fibrotic scarring, and this scarring can actually reduce their mobility. There are a number of treatments that have been proposed, but most of them work primarily for small areas. We're interested in being able to address larger area kinds of wounds. So in this work, it's sort of summarized in this slide, we looked at the in vivo knockdown of one of the growth factors in the pathway of fibrotic response. This is connective tissue growth factor. Here we're trying to address a different part of the wound healing process. In chronic wound healing, we had the difficulty of actually getting the wound healing process started, and we needed to get collagen production to begin. Here we have the opposite problem, a kind of overhealing. In this fibrotic stage, the deposition of collagen is accelerated, and we can't stop it. So we're trying to knock down CTGF so that we can lower that fibrotic response. And what we're looking for here is essentially, this is a third-degree burn model that is carried out at the Shriners Hospital, Shriners Burn Center at Mass General Hospital with Martin Yarmish. And in this burn model, you can suture the edges of the burn and then watch to see whether or not the burn contracts. If this tissue contracts the way it shows here, this contractile wound healing is a part of the fibrotic response. In our system, you can see that with just a control, we get the same kind of shrinkage of the wound. But with the knockdown of CTGF in these cases, we can see that there is a real reduction in this kind of contraction. What's interesting here is that we didn't coat an entire bandage. We only coated the suture. We're actually literally coating the thread that is used to sew up the animal, and we're seeing that there is a meaningful response to the siRNA that is being released from that suture. So we think that the application of this approach is very promising because we can control the deposition of our multilayers on a number of different surfaces that might be used in this wound healing application. It's a little difficult to see here, but we did look at some of the characteristics of scarring, and what we found was that for our SI-CTGF systems, we increased vasculature, and it's a little hard to see, but these are blood vessels where the black arrows are, compared to our scar tissue, which shows very few, if any, blood vessels. And we also see an increase in the size of the blood vessels. We also had a much rougher epithelial surface, and the indication of structures forming that are essentially similar to what we would expect for the formation of hair follicles or sweat glands. So we think that this is the beginning of some very exciting work in which we may be able to address fibrotic scarring using a very rational approach through the delivery of controlled factors and siRNA. All right, so we've moved through two examples in which we've talked about wound healing, and we've gone from bone, which is, you can imagine something you can hold in your hand and imagine, to these microscale meshes, which we can coat very readily. And I'm going to talk about coating something on the nanoscale. And in this work, we're working at the Koch Institute with engineers and physicists and chemists, biologists and clinicians to address cancer. And what's exciting about this work is that we can use a very modular platform to integrate a rational approach to cancer. The idea here is that we can take a nanoparticle core, and that core can be any core. In our case, we're interested in vessels that have already been FDA approved. So it could be a PLGA nanoparticle, it could be a liposome that has a negative charge. And into that core, we can incorporate a chemotherapy drug. We're very interested in highly aggressive forms of cancer that recur or that are very resistant, even on the first treatment, to the chemotherapy drug. A number of these aggressive forms of cancer have undergone the kinds of genetic modifications that allow them to encode proteins that save them from the impact of the chemotherapy drug. So the idea here is that we can incorporate into the outer layers siRNA or perhaps an inhibitor that will block that pathway for cell survival in the presence of the chemotherapy drug. Finally, we rely heavily on the fact that we're using a polyelectrolyte approach because this last layer that's put down is a highly negatively charged layer, and that's going to provide us with a nanoparticle that is much less likely to engage with other cells when we introduce this into the bloodstream. And I'll talk a little bit more about the role of that outer layer. So this is the general idea. Making it nano-sized helps us because in cancer therapies, we're trying to take advantage of the fact that solid tumors, or at least a large number of solid tumors, grow very rapidly. In that rapid growth, they generate defective vasculature. And the defects or holes in those blood vessels provide us a route for nanoparticles to essentially extravasate or enter the tumor tissue and give us an additional residence time in that tumor. So I showed you the slide with the beakers. You're probably thinking, what are you going to do with nanoparticles to get these nanolayers on the surfaces? In our work, we can use an alternating titration adsorption step with a centrifugation step. We're beginning to replace the centrifugation step with a hollow fiber filtration step, which is turning out to be very fruitful for us and also speeds up the rate of generation of these nanoparticles significantly. I have very happy students right now because of that. Now, this is showing you a quantum dot, a lead sulfide quantum dot that had a net negative charge. And what happens is we begin to put layers down on that quantum dot. We chose this because it's small, it's 20 nanometers, and you can actually see the impact of each layer on the size of the nanoparticle. And you're seeing that you're adding anywhere from 5 to 10 nanometers per absorption step. You can also see the charge going from plus to minus as you build up the layer-by-layer film in these nanoparticles. When we first began to look at this work, we chose some very simple polymers to coat with. We used poly-L-lysine and poly-L-arginine as friendly polypeptides, and we used a number of the glycosaminoglycan family as our negatively charged system. And I'm describing some of the work of Stephen Morton, who is a graduate student in my lab. When we began to examine the behavior of these systems, we made several interesting observations. The fact that this is a layer-by-layer film gives us an advantage because we can layer a film which has a very stable nature to it at pH 7.4. However, with hyaluronic acid, it turns out, this particle built at pH 7.4 gives us this strong negative charge, which begins to disappear if we move it to slightly more acidic environments. Because what's happening is we're beginning to protonate those carboxylic acid groups. And as we do that, we're beginning to erase the negative charge. The net impact of that is twofold. One of them is that if you erase the negative charge, you're erasing some of the electrostatic interactions, and the film's cross-linking decreases and it begins to swell. Now, rather than this compact, negatively charged halo, we have this big, fat, but rather soft, swollen layer around the nanoparticle. The second thing that changes is the net charge. It goes from minus 30 millivolts to almost plus 4 millivolts. Its charge shift is significant because we were relying on the fact that it was strongly negatively charged as one of the mechanisms to avoid interactions with cells. On top of that, we were relying on the fact that hyaluronic acid is highly hydrated. So we believe that these nanoparticles have a large amount of bound water around them, and it's that bound water shell which is helping us to repel the absorption of proteins in the bloodstream. And that gives us a long residence time in blood. So the interesting thing here is that with these two changes, we see a huge increase in cell uptake, non-specific cell uptake. This means that if we're able to enter a tumor that is hypoxic, we may be able to enhance the uptake in hypoxic tumors based solely on the physical-chemical properties of the layer-by-layer film. So we tested this, and we found that when we injected animals with solid tumors and looked at where those nanoparticles localized, they co-localized strongly with hypoxic regions of the tumor. Here we used HIF-1 alpha as a way of labeling these hypoxic regions. The nanoparticles are labeled red, and you can see this overlap in this yellow zone here. However, getting into the tumor is a good thing, and encouraging uptake through these nonspecific interactions can also be good, but we can't rely only on hypoxia. Tumors are highly heterogeneous. We need a way to ensure that these nanoparticles are taken up by tumor cells. And in this case, we decided to use molecular targeting. In our systems, we can attach a range of different ligands, from folate to very specific binding peptides that bind to receptors that are overexpressed on cancer cells. And we have indeed done that with a number of our layer-by-layer systems. But it turned out that because we were focused on ovarian, high-grade ovarian cancer, triple-negative breast cancer, and some of the advanced lung cancers, all of those cancers overexpress the CD44 receptor, which happens to be the hyaluronin receptor. As it turns out, that high affinity for CD44 allowed us to use just the hyaluronic acid outer layer as the targeting molecule. So once again, we did a series of experiments to show that we're getting targeting based on CD44 interactions with hyaluronic acid. And I'm describing the work of my postdoc, Eric Drayden. In this work, we looked at xenografts of triple-negative breast cancer, and we did show that these overexpress CD44. When we look at the uptake of these nanoparticles labeled red in this image here, we see a huge amount of uptake, about 15 times that of a layer-by-layer particle that contains dextran sulfate, similar charge, no hyaluronic acid. And we can also titrate out the interaction by adding excess hyaluronic acid, and here you can see that almost completely eliminates the uptake. When we do a tumor section in this case and label CD44 with red and the nanoparticles with green, again we can see a huge overlap between the localization of our nanoparticles and the localization of the tumor cells. And what we see in this outer region are the stromal cells that surround that tumor, which suggests that we have a mode of triple targeting. We're using nanoparticle size, we're using the tumor microenvironment, or hypoxia, and we're using the cell receptor interaction to ensure that these nanoparticles are taken up. Now that we have a system that can do some form of targeting, we can load the inner core of the nanoparticle with a chemotherapy drug, and we can begin to layer our siRNA. We actually found that the positively charged polycation that we use in that layering process greatly impacts whether the siRNA gets both into the cell and out into the cytosol. It turns out that polyelarginine is a polymer that undergoes a great deal of buffering and therefore enables these nanoparticles to essentially compromise the endosomal membrane, and that allows siRNA to get out. So what we began to develop then was a 4-layer nanoparticle. We would start with a core nanoparticle, and then we'd layer polyelarginine, siRNA, polyelarginine, and hyaluronic acid, plus, minus, and that would be our outer layer system. we found that when we generated these nanoparticles, we got very nice blood half-lives. This is key because we need a long residence time in the bloodstream to be able to allow nanoparticles to accumulate in the tumor. In this case, we found that we had a 28-hour half-life, which is actually quite extended for these kinds of nanoparticles. The size of these nanoparticles are 120 nanometers. I should also mention that we didn't observe any kind of immuno or cytotoxicity. We did, of course, use a fairly simple panel of cytokines to examine that. If you look at the release characteristics of the nanoparticle, once again, we see the staggered effect. We can release siRNA over a more rapid time period of about 24 hours. In this case, we load our liposome with doxorubicin. So we have this co-delivery. We're using a negatively charged phospholipid liposome. Doxorubicin is water-soluble and goes into the interior of this liposome, and siRNA goes on the outside, and we get our stagger. Our siRNA was siRNA against multi-resistance protein 1, or MRP1, which is a drug pump that pumps out doxorubicin even as it arrives at the cell membrane surface, thus preventing it from interacting or engaging the nucleus and killing tumor cells. In this work by Jason Dane, who was a postdoc in my lab, we looked at a series of injections in these animals, which contained xenografts of a triple-negative breast cancer. We found that we were able to get significant knockdown after treatment. Here we're looking at tumor sections that were stained, and here we're looking at PCR, which shows that there is a roughly 75 to 80% knockdown of MRP1 in these tumors. When we examined using a xenograft model tumor remediation in these animals, we find that in the saline control, we're getting, of course, this rapid growth of the triple-negative breast cancer. If we look at doxorubicin with the scrambled siRNA, we see that we are reducing the growth of these tumors, but not their size. They're still essentially doubling in size. However, if we treat these with the combination system, we do see that the tumor size is getting smaller, and we're getting some real regression. So we're very excited about these results, and we began to move to a more challenging model. Tyler Jacks at the Koch Institute has developed a model in which these mice are essentially genetically modified so that they have mutations in KRAS and in P53. And I won't have time to go into the details, but these are two of the highly known oncogenes. Inhibition of KRAS, therefore, is highly desirable, but small molecules have not been developed to effectively inhibit this KRAS gene. It's also known that P53, which is one of the guardian genes that prevents the kinds of tumorigenic activity that we see in cancer cells, is also lost. So we're going to introduce a microRNA to restore the P53 function in these cells. Again, we're going to build up a 4-layer nanoparticle, as shown here, but this time with cisplatin, and the two different RNAs, siRNA against the KRAS oncogene, and microRNA miR34. And here we're just describing this orthotopic KRAS mutant model. I won't go into the details, but what it does allow is for us to activate tumors in mice, and these tumors are small tumors spread all the way across the lungs. We looked at the active targeting of our nanoparticle in healthy mice. So here we do a tail vein injection, and we see that the nanoparticle resides throughout the mouse's body, but there's no particular localization, versus mice which exhibited these lung cancers, these mini tumors. We saw that over a period of time, we got a large accumulation of the nanoparticles in the tumored lungs. And just to see the relative differences, here you can see the lungs of the healthy mice versus the lungs that contain these tumors. And here you can see the relative difference in recovered fluorescence. And we're looking at orders of magnitude difference in the amount of accumulation of these nanoparticles in the lung. And this is due to the fact that, again, CD44 is highly overexpressed in this lung cancer type. In our in vivo treatment, we used, again, this orthotopic model. So it's a little bit hard to see the tiny tumors on this screen. However, you can look at the fold change in tumor volume here and see the vehicle versus RNA. cisplatin, and the combination. And we see that there is a very meaningful difference when we look at the combination therapy, both in survivability and in fold change of tumor volume. We also can check and see whether or not we have modified the pathways we think we're modifying. And we see from this series of staining against pERK and a range of other proteins that we have downregulated the KRAS pathway, that we have reinstituted the p53 pathways, and that we have enabled cell death through caspase-3. So I'm coming to the end of my talk. I'm going to give you a very short example. This is a microsecond extension of our nanoparticle project, and I'm bringing it up because one of the exciting things about working at MIT and at the Koch Institute is being able to work across disciplines. A lot of the work I described previously has benefited from the collaborations of Michael Yaffe, who's a systems biologist, and a range of clinical researchers at Dana-Farber and Mass General Hospital. I also have a collaborator who's another engineer, Angie Belcher, and she engineers a range of different inorganic molecular systems using biological methods. She has, among her innovations, been looking at ways to develop nanoparticles that absorb in the second window near IR. This is a window of near IR we haven't taken advantage of as well, but would allow us to get much deeper penetration for imaging of tumors. And in this work, my postdoc, Lee, and Angie's postdoc, Janan, have worked together to see if we can develop an imaging nanoparticle in which the core contains one of Angie's down conversion nanoparticles, and the outer layers contain an LBL film, which could ostensibly contain siRNA, plus a range of inhibitors or chemotherapy agents. So we actually looked at a range of different layer-by-layer systems to compare with Angie's quantum dot and down conversion nanoparticle. And this is just showing some of those characteristics and images of those particles. And what we found is that by using Angie's system, in comparison with the others, we can completely escape a range of autofluorescence that we experience with dye molecules and single-wall nanotubes, and actually see what's going on inside of these animals without that autofluorescence using the down conversion nanoparticle and quantum dot. In fact, with the down conversion nanoparticle, we can see fine structure of blood vessels, musculoskeletal structure, and even the beating of the heart is much more visible. So we've been able to apply this to the imaging of ovarian cancer tumors by using that hyaluronic acid binding capability. And here we've shown in an orthotopic ovarian cancer model that we get a huge amount of sort of concentration of these nanoparticles in the regions where we have the tumor. And here you can see just images that are sections in which we compare the nanoparticle accumulated in tumor versus healthy tissue. And you can get down to sub-millimeter scale resolution with these systems and actually see and differentiate the tissue between tumor and pancreas, gut, liver, and of course we get a very nice view of solid tumor tissues. So right now we're developing this technology to see if we can generate these kinds of theranostic systems in which we can monitor ovarian tumor suppression at the same time that we're delivering therapies. So in conclusion, I talked about the fact that we can use this very modular, water-based approach to incorporate a range of different molecules, and that they can be used for very significant wound healing applications, including the cranial defect example. I didn't talk about, but we've also had collaborations with members of the Koch Institute, including Daryl Irvin, on the incorporation of layer-by-layer films for vaccines. And I talked about how we can use these kinds of layer-by-layer films, nanofilms, on the nanoscale and generate very effective targeting nanoparticles. I think this is a very modular approach, and this is why we're excited about it. It can be applied to a very broad range of surfaces, and it can also be adapted for use with a range of other kinds of tools for health applications. So what we're showing is that electrostatics is a very critical tool that can be used for controlled release, and that this approach can introduce functionality that can lead to very promising biomedical applications. I'd like to thank the contributors to the work. I've highlighted my students and postdocs in bold, whose work I highlighted here, as well as my collaborators, Myron Spector in a lot of our bone work, Michael Yaffe, who's the biologist at the Koch Institute we've worked with, Angela Belcher. Right now we're working with Mike Beerer at Mass General Hospital to address some of our ovarian cancer applications. And I'd like to acknowledge the NIH for funding a number of these early discoveries in our lab and for continuing to support our work. And I'd like to thank the Ovarian Cancer Research Program and Research Foundation. And welcome any questions that you may have. Thank you.
Thanks very much for a fascinating romp through a lot of applications. And please, if people have questions, come to the microphone so those watching can hear. Please start here.
Thank you very much for a wonderful presentation. What I was interested in understand is how consistent is the loading of your experimental, your actual therapeutic agents into either the films or the nanoparticles, and what level of variation you tolerate. And as we really want to see this moving forward into a clinical, what do we think the FDA is going to say about that? How consistent do they need to be?
Excellent. Actually, they're extremely consistent. One of the nice things about this polyelectrolyte approach is that charge is very predictable. So if you assemble these systems under the same conditions, you end up with the same amount absorbed. If we look at our, you saw several curves in which we were building films, and you see these linear curves. That linearity is beautiful, and it's always there. So we can go back to the same conditions and generate the same films. This has been shown by the fact that, for example, in the bone work, we've passed on this formulation from across 3 students and a postdoc, different hands, same loading. And I think this will help us with the FDA, that we can actually use this as a very reproducible effect. You have to do something quite intentional to disrupt the sort of electrostatic adsorption behavior of these systems.
So that's called a biological replicate when it's more than one postdoc doing the same experiment, I guess.
I like that.
What happens to these nanoparticles that you've put into the body? Where do they go? How do they ever disappear?
Very good question. Although they have a very long half-life, they will accumulate in the liver and the kidney, and this is why we've been trying to measure signs of distress. We measure body weight, and we also measure cytokines to see if there's any inflammatory response or any other kind of response. We've also taken out the liver and resected it to see if there's any long-term accumulation in the liver. What we find is that they pass out of the liver, so if you wait roughly 24 hours to 36 hours later, you're not seeing these nanoparticles in the liver anymore. So over a period of time, they're actually somehow breaking down and getting out.
So they are biodegradable. They're not.
They are. Now, that is a good point. Each of the systems that we choose for the nanoparticle applications contains degradable components. So we're using a phospholipid liposome and then layering it with these hyaluronic acid containing systems. And so we try to maintain a system that's going to either be resorbed or broken down.
Got it. Over here.
Well, congratulations for pushing the difficult frontiers of nanomedicine. So in one of the studies recently showed that the talc powder causes cervical cancer. In fact, it was recently settled. So as you are putting the layers, you also have to be careful which might be the acceptable model versus which could get you into trouble. So in terms of multilayered particles, I was looking at it. You had the layer of hyaluronic acid. Is there any pegylation to prevent phagocytic removal of the nanoparticles?
Excellent. We actually considered PEG versus the hyaluronic outer layer. And there's a couple of papers that you'll find of ours in which we capped off the final layer with a PEG brush. We actually find that the half-lives are very similar or better for the hyaluronic system. And there are reasons for this, and in fact, there are other research labs who have used hyaluronic acid as well, and they're beginning to use it more frequently. And in fact, Sean Shin is one of the labs that has looked at this to some extent. But we think there are several reasons. One of them is that hyaluronic acid itself has a great relationship with water. And for that reason, that bound water is helping us with the issue of both steric repulsion. It provides some steric repulsion, but it also provides a bit of a cloaking layer behavior. It prevents opsonization, so proteins tend not to absorb as readily on these surfaces. But we also think that the presence of the layer-by-layer film has an influence. So when this absorption takes place, the polyelectrolyte is absorbing on a positively charged underlying surface, but it's not a flat surface, it's another coiled polymer chain. And we end up getting these loopy structures that absorb. So the molecular conformation of the polymer chain is a little bit different. We think that we have this brush-like structure formed from these loops, which is actually providing a much denser cloud of water and associated ions than you would get if you had just an extended linear chain extending from the surface of the polymer, of the nanoparticle.
Over here.
Hi, it's a very interesting talk. Polysaccharide, as you know, is a very common vaccine component. There are at least 23 different in pneumococcus, there are 23, and they are all licensed. In meningococcal, there are 4, and Haemophilus influenza. They are mostly negatively charged, you probably know. Have you thought of using any of them, the vaccines, as your coating, as your layer?
Ah, very interesting.
So that would be, like delivering the vaccine as well as for other purposes too.
Excellent. In fact, I had a very interesting conversation today with Joe Barsky, with someone who is actually working on glycoholic materials. And he talked about the fact that he can create glycopeptides that are highly immunogenic. This is actually very interesting to us because we think we can combine those kinds of interactions with our layer-by-layer system, initiate an immune response, and then release an antigen and have control of the rate of that presentation of antigen. And that's interesting to us. We have a collaborator, Daryl Irvin, and we've begun to talk about using our layer-by-layer approach for immunotherapies for cancer. And we have our first student who's going to be working on this. So these kinds of ideas are very helpful at this stage. I do think it's interesting. What we've learned from the microneedle work I didn't talk as much about was that we can coat a nucleic acid that encodes for these or coat a protein onto these layer-by-layer films, get a very slow release, and for various reasons, getting the slower release of the antigen seems to give us a more sustained memory T-cell effect. And that may be something that we can use for immunotherapy against cancer, if we can get this more sustained release.
Yeah, the licensed vaccine can come in 2 forms. One is the polysaccharide alone, and the other one is a chemical conjugate into the protein. I have another question. That is one of the most common negatively charged polyelectrolyte is nucleic acid. Have you ever thought of using polynucleic acids?
Could you say it just one more time?
Polynucleic acids.
Oh, polynucleic acids.
Right.
Yes.
They are very regularly negatively charged.
We have, so ironically, or coincidentally, we have a project in which we have been generating very large hairpin-shaped RNA. Essentially, it's a kind of multi-periodic RNA structure. And that work was really directed toward a different part or different approach to siRNA therapy because these break down in the presence of dicer into hairpins that can be used for delivery. But we're discovering that those structures are activating the NCAPA beta mechanism in cells, and we're actually getting an inflammatory response that seems specific or at least highly amplified in cancer cells. So what you're saying is, I think what we're beginning to observe with these molecules, that's very interesting.
So recognizing that we are a bit after the hour, and I know people have things they need to do, maybe others who have questions can pose them to our speaker as we all migrate towards the library for a reception with some coffee and cookies. But please join me in thanking Dr. Hammond again.